All right, so we talked about microevolution. Now let's talk about what a population is. I just mentioned population a bunch of times when I was talking about the frogs. So a population is any interbreeding group of the same species. The idea is when there's a bunch of individuals of a species in one area that are able to reproduce with each other, we call that a population. So why is a population significant when we're talking about evolution? Well, an individual doesn't evolve, right? From the time an individual is born to the time it dies, it has the same genes the whole time. A population, on the other hand, has this collection of genes. It has the gene pool. And so over time, from generation to generation, the frequencies, the allele frequencies, will change in a population. And so evolution occurs at a population level. But when we talk about natural selection, when we talk about a predator taking an organism out of the gene pool, or a female of a species only allowing certain individuals to pass their genes along in a gene pool, that occurs at an individual level. So this is kind of an important distinction between natural selection and evolution. Natural selection occurs with individuals, while evolution is a trend that occurs in populations over time and over generations. There's a way of measuring that, and there's a way of looking at that, and we call that the Hardy-Weinberg equilibrium. Basically, this is two equations, and each one relates to a different component of either allele frequency or genotype frequency. So if you remember, the genotype was the combination of the two alleles. When we said that something was homozygous dominant, that was the genotype. It was two of the dominant allele. When we said something was heterozygous, we were talking about an individual that had one of each kind of allele. And when we said something was homozygous recessive, that had the two recessive alleles. And that was with the Mendelian gene. In any population, you're not going to have a 50-50 split of each one of those alleles. Remember, we're looking at alleles as a gene pool. We're no longer looking at genetic information from the individual level when we're talking about allele frequencies. We're just looking at the gene pool. So a population could have 60% of one allele, let's say the dominant allele, and then 40% of the other allele. And the interesting thing that happens, and this is what the equilibrium part of the Hardy-Weinberg equilibrium is referring to, is that if there's no natural selection happening, there's no selective force influencing this population, then that ratio, that'll stay consistent and constant through time. So they call that an equilibrium because it's balanced. It stays that way. But first, let's look at the Hardy-Weinberg equilibrium. And we're going to first break it down into its components and figure out why it makes sense. Then I'll show you an example with a Punnett square. Then I'm going to write down an example for you that I just made up. Then I'm going to have you do your own example. And this will be likely the most in-depth math I'm going to have you do in the whole semester. But I think it's important. So, the Hardy-Weinberg equilibrium. This is actually a pair of equations. It's two equations. And the first one is referring to the alleles. In this case, the sum of the alleles has to equal 1 because 1 is the total. So here's the frequency of one allele, that's P, and the frequency of the other allele is Q. It equals 1. No matter how you divide up the population of alleles, when you know what one value is, you'll know the other one because it always equals the sum. So with alleles, it's pretty simple, pretty straightforward. The part where it might get confusing is when we talk about the genotypes. So remember, we did a Punnett square, and we had homozygous dominant, homozygous recessive, and heterozygotes, right? Those are the genotypes. So that's the combination of alleles an individual has. This first equation, P plus Q equals 1, that's for alleles, not genotypes. That's just for the total kinds of alleles in the total gene pool, not thinking about the individual organisms. when we're thinking about genotypes, we are thinking about the organisms, and we're thinking about what their particular combination of alleles are in that population. So if you look at the equation for genotypes, the Hardy-Weinberg equilibrium equation for genotypes, the frequency of one set of homozygotes is p squared. The frequency of heterozygotes is 2pq, and the frequency of the other set of homozygotes is q squared. These aren't referring to whether these are the dominant or recessive homozygotes. It's just saying one set of the homozygotes is going to be p squared. The alternate version of that, the alternate allele homozygote will be q squared, and then the combination of those two in the heterozygote population will be 2pq. So the second equation, this equation for genotypes, this is talking about individuals. So this is saying the number of these homozygote individuals plus this number of heterozygote individuals plus these guys, that's always going to equal 1. That's your total. It will be 1. Because, of course, they all add up, make your entire population. So how is that helpful? Let's look at an example. So in this example, we have a Mendelian gene, and we have a dominant allele. We'll say that's R, big R. Big R is the dominant allele. And then we'll say little r is the recessive allele. Well, in this population, if we weren't to look at the individuals, but we were just to look at the frequencies of alleles, big R has a frequency of 0.8. 80% of this population has big R. If you look at little r, it's 0.2. 20% of the population has the recessive allele. But we don't know what the genotypes are yet. We just know that that's the frequency of this allele in the population. So let's go back to a Punnett square. And here we're going to say that this R allele, big R, little r, is expressing color in a flower. In this case, big R is a dominant allele, and it expresses a red color in a flower. Little r is not expressing the red, and we'll say if you have homozygous recessive with little r, the flower color is white. So this was a cross we did before. So if we ignore all the values here, the cross is very familiar, right? The heterozygote gametes for the maternal side are big R, little r, right? Because it's heterozygote, so the maternal side has both big R and little r, and so the gametes have big R, little r. The paternal zygotes are also big R, little r, because those are also heterozygote gametes, right? The gametes from a heterozygote. And when you do the cross, you get this ratio, right? You get one homozygous dominant, big R, big R. You get a big R, little r here, a big R, little r here, and then one homozygous recessive. So you get 1, 2, 1, right? One homozygous dominant, one homozygous recessive. You get two heterozygotes. So now let's do that same cross, but instead of using the allele names, big R, little r, we're going to add their values, and we're going to do the same cross, right? So now big R, we know that that has the frequency of 0.8, so we're going to give big R 0.8. Over here, the paternal big R, we're also going to give that 0.8. And when we do that cross, we're just going to multiply those two, 0.8 times 0.8, and that equals 0.64. That's also the same as 0.8 squared, so that's p squared. Or you could think of it as r squared. However you want to think about it, it's this value times this value, which is a squaring of that value. So p squared, r squared, whatever, it's 0.8 squared in this case. Now we'll do this cross with little r and big R. So little r has a different allele frequency. Remember, it's 0.2. So we're going to multiply 0.2 times 0.8, p times q, and that equals 0.16. It's not so hard, right? Remember this heterozygote, there's another heterozygote over here, and we're also going to do the same thing, p times q. That's 0.8 times 0.2 equals 0.16. Well, because there's two of these, we can write these down as 2pq. This is p squared, remember, because we're multiplying big R. It's squaring of these two. And then finally we have the recessive allele. 0.2 times the other recessive allele, 0.2, equals 0.04. So this would be the same thing as writing q squared or little r squared, but it's these recessive alleles squared. So now instead of just having the F1 frequency, we've added in the values of those frequencies in the populations and actually gotten these frequencies for the genotypes. So if this was a population of 100 individuals, let's say, the allele frequency would be 80% big R and 20% little r. But what does that look like as far as genotypes? Well, we just did it with this Punnett square, and the results are this. Your big R are occurring at 0.64%, right? So that's in a population of 100, that would be 64 individuals that are red and are homozygous dominant. In this population of 100 individuals, your heterozygotes, 2pq, would be 0.32. So you would have 32 individuals that look red, but they're actually carrying that recessive allele. So those are your heterozygotes. And then your recessive allele would be at 0.04, which is 0.2 times 0.2 would be 0.04. That would be your genotype for the recessive allele, your homozygous recessive. So you would expect to find 4 white flowers, which is not many. But you can see there's actually quite a few individuals in this population carrying that recessive allele. Let's just say we have a population of little students, and we know this about these little students. Some of them have devil horns, and we know that that's recessive. And we know that there's some mystery number of dominant, homozygous dominant, and heterozygotes in this population. We don't know what these are, though. We don't know what they are, but we do know that we see 0.01. Out of the 100 students, one of them has the devil horns. That's the genotype that we're seeing, right? And this is genotype. So we don't know what the other ones are. So we have two equations to work with here. We have allele frequencies, and that is P plus Q equals 1. Then we have genotype, and that is P squared plus 2PQ equals, get rid of that right there, plus Q squared equals 1. So between these two, we should be able to figure out these other two genotype frequencies. So this is a genotype here. This is a genotype. So to figure out what that is, we have to use this equation. And we know that both of these frequencies, their homozygous states, equal P squared and Q squared. So we know that this is going to be, GG is going to equal, let's say it's P squared, and that equals 0.01. So if that's the case, the square root of P squared should equal P. So the square root of 0.01 should also equal P, because that is P squared, right? So the square root of 0.01 is 0.1. So that means that's equal to P. So now we can plug this in to the allele frequency equation, and we get 0.1 plus Q equals 1. So if we subtract 0.1 from each side, we get Q equals 0.9. So now we know that Q equals 0.9 and P equals 0.1. Remember, this is allele frequencies now. We figured out the allele frequencies. Now that we figured out the allele frequencies, we can figure out the genotype frequencies. So now let's plug these values into the genotype frequencies to understand what they are. P squared. Remember, this is this value here. P squared, we know that equals 0.01. And we know that also because our example started out with P squared. 2PQ is going to equal 2 times 0.01 times 0.9. And that equals 0.18. And now we need Q squared, and that is 0.9 squared, which equals to 0.81. So this here, this is your frequency for your homozygous dominant. This is your frequency for your heterozygotes, and this is your frequency for your homozygous recessive. So now we can get rid of these question marks and fill it in with the information we just made or just figured out. We know this frequency is 0.81 and this frequency here is 0.18. So within this population of 100 students, we have one that expresses the recessive, homozygous recessive state, and that individual has devil horns. But in that population of 100, we'd expect 18 to be heterozygotes carrying the recessive allele. And then we'd expect 81 of those students would be homozygous dominant. And that should be stable through time unless some external force is acting on that equilibrium. This uses these two equations, and the only thing that is tricky about this is keeping in mind whether you're talking about a genotype frequency or a genotype or an allele frequency. So when I was starting out with the genotype here. I used that, and I found the square root of that to find the allele frequency. And once I had this allele frequency here, I was able to then plug it in to the broader equation and figure out all the other frequencies. So now's your chance to do exactly that. It might take you a couple tries, but I'm going to give you some values, and I'd like you to find the missing genotype frequencies and allele frequencies for a problem of your own. So by now you understand that there's a relationship between the frequency of alleles in a population, and that tends to stay pretty stable unless something's changing it, unless there's a force acting to change that frequency. In this example, the frequency is 0.8 and 0.2. Well, without evolution and without any other forces, you should expect that to stay the same generation to generation. It's kind of weird to think about, but you know, if you had dice that had all but one side one way and then one side another way, you'd expect that ratio of rolling the dice to stay about the same no matter how many times you rolled it.